özellikle sorumlu.
Esteemed district governors, honorable rector, distinguished academics, consular, respected representatives of the civil society, and dear students. On behalf of the Gebze Technical University, it's a great honor to welcome to you all. Hoş geldiniz. Today is a day we will remember for a long time. At Gebze Technical University, we have strong foundations in physical sciences, as well as in life sciences, architecture, engineering, and social sciences, and you know, all these disciplines that complement each other in addressing real-world challenges. With our unique position at the intersection of academia and industry, we aim to advance knowledge and create meaningful impact through contemporary research-focused work for everyone. Our chemistry-related fields stand among the most competitive globally, with chemistry ranked within the 500 and chemical engineering within the type 400 worldwide, reflecting our commitment to high-quality, impactful science and international excellence. We should remember that awareness for science should begin very early, because it's a natural and strong way to understand entire life. Sometimes in a kitchen, sometimes in nature, it starts with simple observations and questions and can lead to discoveries that transform entire disciplines. Without fear, asking questions and remaining open to learning can change many of challenges we want to solve in every day, like Professor Meldal indicated. Today, we are honored to welcome a scientist whose work has fundamentally reshaped the way we construct and understand chemical reactions. Professor Morten Meldal was awarded Nobel Prize in Chemistry in 2022 for the development of Klik chemistry and bioorthogonal chemistry. It's a great honor for us to host a scientist work has changed our life in interdisciplinary way to solve different problems in every day. So it's also a great honor for us to once again welcome a Nobel laureate under this building, under this Gebze Technical University, following our distinguished previous guests, Aziz Sancar and Peter Grünberg, as part of this continuation of the tradition. Before proceeding the introduction of our distinguished speaker, I would like to first thank the organizing committee from the Faculty of Sciences and the Department of Chemistry, as well as our sponsor for being here and gathering under this meeting. And so the Dr. Erhan Atıcı representing the Chemists Association, Kimyagerler Derneği. So we sincerely appreciate the valuable support of Kimyagerler Derneği because their contribution plays an important role in promoting science communication and strengthening the connection between scientific knowledge and society. For this reason, first, I would like to now invite Dr. Atıcı to the stage.
Dear President and the Consulate of the Denmark and the professors, scientists, colleagues, students, especially our honored guest, Professor Meldal. It's a great honor for us and pleasure to welcome to you. And we are here, many people here. We are gathered here under the power of the science. Since its establishment, our society, Turkish Chemical Society, Chemist Society, has supported the mission of the contributing to chemical industry and academy of the chemicals. We are performing many ceremonies and organizing major congress during the years, and we are supporting the students at their first day of their life with the white coat ceremonies. And also, we are also organizing career talks to support them, to guide them, what they will do after graduation. With our congress, it is international and national congress, so with all these things, we think that we create a very solid scientific area with our society. So that's why we are here. We want to share our knowledge together, and we want to inspire our special guests. Science is a journey with curiosity and grows with patience. Patience supports the growing. But it gets meaning when you serve to humanity. So chemistry in particular, it is one of the most transformative pillars for this area. It is presence of chemistry. We can feel every point of our life. It touches every point, from automotives, from aeroscopes, from energy, from mining, from pharmaceuticals. You can feel the chemistry every point of your life. So our contribution to chemistry, in fact, it is the investment in future of the human. We believe that. So we know our responsibility for this point. We are aware of this one. So we not only follow the science, we also sharing, we are producing, and we are developing. Supporting our young colleagues and encouraging them, guiding them, our main responsibility also for this purpose. Today we have a very special honor. We have hosting truly a very special scientist together with us, Nobel laureate Mr. Meldal. We will learn many things from him. We will learn their pioneering works. Although he has a very busy schedule, he gave me appointment for 20 months later, but at the end he is together with us. Thank you again. We will learn many things. We will share our knowledge. We will learn his experiences here, especially for the young scientists. It will be very helpful, I think. So I want to express my sincere regards to him one more time. Yes. Because we believe that if there is science, there is future. Where there is chemistry, there is transformation. So all of people's here, so I think we have hope. We know the situation of the world these days. So because of these peoples, I think we have hope. To sum up, in closing, I would like to share my sincere regards to Mr. Meldal one more time. And to all, thank you for welcoming here, and thank you for listening. Thank you.
Welcome to the Nobel Talks, proudly presented by Turkish Chemist Society and Gebze Technical University. A mind that reconstructed our world at the molecular level, redefining the very boundaries of science. 2022 Nobel laureate in chemistry, Professor Morten Meldal. His journey did not begin in state-of-the-art facilities, but rather with a pure, unfiltered curiosity about the natural world on a small island.
I was very lucky because my grandparents have a farm in Bornholm where I was shipped off to every single opportunity they had. So I was there, and I was out in the morning at 8 and in at 9 o'clock in the evening. So I had a whole day of nature and being in the fields, in the forest, in the beaches and so on. It was really nice as a child.
That drive to understand nature's kinetics soon evolved into a passionate and sometimes explosive interest in chemical reactions.
Later on, when I was 16, I ventured out to a small company that sold all sorts of chemicals and bought what you would consider today to be explosives. But eventually I made something that exploded in the backyard of my parents' house, and they actually sort of stopped the exercise at that point. But it created a lot of interest in me for understanding the chemistry, how it works, the reactions, the kinetics, and why is it that chemistry actually happens.
That teenage curiosity eventually triggered a revolution in modern science. Professor Meldal is a pioneer of Klik chemistry. Traditional chemical synthesis is often slow, complex, and generates unwanted byproducts. Professor Meldal engineered a flawless alternative. By introducing copper as a catalyst, he discovered a method to bind two specific molecular groups, azides and alkynes, with absolute precision and instantaneous stability. This reaction is exceptionally highly yielding and specific, resulting in zero chemical waste. Today, Klik chemistry serves as the architectural foundation for groundbreaking advancements. It enables targeted cancer therapies, precise DNA mapping, and the synthesis of advanced industrial materials. It is the epitome of pure, efficient science. For rendering complex chemical reactions entirely controllable and pure, he was awarded the highest honor in science, the 2022 Nobel Prize in Chemistry.
When I received the message from Stockholm on the Nobel Prize, that was really cool. First I thought it's probably a prank played by my students, because I've tried that before. But I could very, very quickly hear that this was not a prank and this was serious. And I talked with 3 committee members, and they told me about it, and it was very, very exciting. Then I had to wait for an hour, and that was kind of tough, to wait for an hour not telling anyone. Even my wife I couldn't call. And after that I heard the announcement, and as soon as I opened my office door, there was like hundreds of people in the corridor outside, 50 people in the corridor outside, all cheering, and that was very, very pleasant experience.
Despite global recognition, Professor Meldal's true driving force remains far from the spotlight. He is an academic at heart, deeply committed to the laboratory and the next generation of scientists.
I actually work still in the laboratory, and I work in the laboratory because I think it's fun. I'm not driven by ambition, I'm not driven by another Nobel Prize. I'm driven because I like to investigate chemistry. I like the understanding of the world that we are living in.
A scientist who proves that true innovation comes from stepping into the unknown. We are profoundly honored to present the Nobel Talks, bringing world-changing ideas to the next generation of researchers. Because the pursuit of knowledge never truly ends. Welcome to the Nobel Talks.
It is a true honor for us also. In the next part of our program, Professor Meldal will deliver his lecture, which will be followed by questions and answers session. And so it's a great pleasure to present first this bio-sketch of Professor Morten Meldal very shortly, who is the 22 Nobel laureate in chemistry. He received the Nobel Prize together with Carl Barry Sharpless and Carolee Bertozzi for the development of Klik chemistry and bioorthogonal chemistry, approaches that have made chemical reactions faster, more efficient, and highly selective, with important applications in medicine, biotechnology, and material science. He completed his Master of Science and PhD in Chemical Engineering at Technical University of Denmark, DTU, and has made significant contributions to peptide chemistry, combinatorial chemistry, and chemical biology. In 2001, he independently developed copper-catalyzed acid-alkyne cycloaddition reaction, which has become one of the most important reactions in modern chemistry. Throughout his career, he has published over 300 scientific papers, holds multiple patents, and has contributed to both academia and biotechnology and all disciplines. Today, his work continues to inspire researchers like us across many disciplines. Please join me in welcoming Professor Morten Meldal.
Just a second.
Preparation.
Hello, GTU. Hello, Turkey. It's a really great pleasure to be here and a true privilege to see so many young people in the audience. So thank you very much for coming to this event. And also thank you to, for the introduction. It was really nice to see my lecture in the shape of a video. So now I can go home. No. When I was in 2022 sitting in my office and preparing slides for the students, suddenly the telephone rings and I'm looking at the telephone and it's a Swedish number. The week before, I had a lot of calls from Sweden, a company that wanted to sell me an instrument, and they were very persistent. So I was sort of a bit irritated on this Swedish number, so I go, yes! And in the other end, there was a complete silence for a while. And then it was, this is a Nobel Prize committee. So I was very silent for a while. And then we started talking, and it was really good pleasure, a fantastic experience to be called by the Nobel Prize committee and told that you are winning the Nobel Prize. So you cannot tell, as was said here in the video as well, you cannot tell anybody for an hour. I went to get a coffee at some point. I passed our head of the institute, and she was looking at me like this, because my smile was, of course, going like that. And she was wondering what was going on. But other than that, you cannot tell, and it was a fantastic thing. And later on, you are invited for a vacation. I'll give you some insight into that in Stockholm, and this is a highly recommended vacation, because they're very good at it in Stockholm. But something is happening here. What is going on? Do you know what that is? Okay. So I can tell a little story in the meantime. This is about, I'll show you later on, but it's a bit about this thing with the explosives. In Denmark at the time, it was possible to go to a company, a small company south of Copenhagen called Struers. And I was about probably 16, maybe 15 years old. And I went with a cardboard box size like this, and I went into the company. I bought aluminum powder, perchlorates, all the sorts of things you can imagine that is reducing or oxidizing. And I went home with this box of explosives in the S train, so the public train. This would, of course, not be possible today. Home in the basement, and then I have today at home like a lab journal this thick with all sorts of prescriptions for making gunpowder. It's really fun to play with, but it's not allowed today, of course, so sorry about that, young students. You will have to miss out on that. Also, we had a fantastic day yesterday at Temu, and this was after the lecture, a lot of questions, and I would love you, young people in particular, to sit and consider what are your essential questions while I give the lecture, so that we can have a really fruitful discussion and questions and answers after the session. Yeah, this is unfortunate. I'm sorry about that. Is it gonna work? Yes, it's back. Okay, so I got this phone call, but so did Barry and Caroline, two good friends of mine that I know from back in 2001. Maybe I should actually do this. Could that be an incompatibility? Yeah, so Barry and Caroline, two good friends of mine, also so Barry and no, in any event, we heard from Erhan that chemistry is transformative and it is influencing everything we have around us. Chemistry is basically seen as a language that describes atoms and molecules, but chemistry is actually atoms and molecules, which means that chemistry is everything around us. We have a language that describes it, but the actual chemistry is everything around us, everything inside us. Everything that happens in this universe is based on molecules and atoms that come together and do what they do. So this means that when you feel tired, when you have hunger, when you feel pain, so on, this is chemical compounds that are going into receptors, are recognized inside the body and are eliciting these feelings. It also means that everything around us is changing rapidly because we are using chemistry at an unprecedented rate to transform the surface of this earth. And this will not be very different in the next 10 years, but on the long term, this will have a very significant influence on what is happening. Life is composed of only 16 atoms. So 16 atoms is what is all that is needed to create life, which is really strange. We have a very complex organism, all of you here, and this is only 16 atoms that come together and does this. So atoms have this inherent ability to build life. Inside the atoms is an inherent ability to form molecules. There's an energy flow that leads to bonds, that leads to molecules come together, aggregate, forms compartments, eventually make cells and replication and so on. And this probably happens millions, if not billions of places out there. Right now, we only know about it here. So we have to be content with that because the distances in the universe is too large that we'll ever talk with anybody else. But the propensity is there. When you have the atoms, put them under conditions that we have here on Earth, they will eventually create life. And the life is, as I said, composed of the atoms. That is protons surrounded by clouds of electrons. And it's, as a chemist, these electrons that we have to understand. So the protons and neutrons, the atom nuclei, are just anchors in space, and it's in the electron clouds that we have all the information that we need to understand as chemists. And this is a three-dimensional world. A three-dimensional world which is very important for us to see as a three-dimensional world. And that's why I had on the first slide that we should actually teach the young people chemistry from the very first grade. Because the young people have this ability to create images in their minds. They are born into this world without a language, and they learn this world, to know this world in 3 dimensions by storing lots and lots of images, creating a mirror of the outside world that eventually becomes their navigation tool. That is why children in first grade are very good with fantasy. They're very good at drawing images and so on. We take that away from them. We try to learn them formulas and reading and writing and so on. But it's the visualization that is really important for the young. That's why it's boring for them to learn things in school. And we need to take that back, give them an animated chemistry world from first grade that will allow them to instinctively develop an understanding of the chemistry that can make the public understand what happens if we heat this Earth by 2 to 3 degrees. What is actually happening there? All the chemistry involved with the evaporation from the oceans, further out, coming down a lot, large belts of desert, turning Gulf streams, ice age coming down. There's so many things going to happen in the future, and there's only one way we can sort of remedy that, and that is by understanding and utilizing the chemistry. So life is all of that coming together in larger nanostructures. Electron waves respond to the environment. So if you take a compound in one solvent and run it in a mass spectrum, you see one spectrum. If you then change to another solvent, you see a different spectrum. What is that? That is because the electron densities around the nuclei are changing depending on which solvent we put the compound into. What does that mean? That means that the environment is actually influencing the electron density around the nuclei. So, in principle, environment is responsible for the quality of the electron density clouds around the nuclei, meaning that every single carbon atom in you is a tiny little bit different from all the other atoms. This is not a way we usually see it as chemists. All carbons are the same, but actually they're not, and that's what leads to the complexity that is you right now. We work with biology, molecular recognition, molecular transformation, molecular signaling, transport. We don't work with DNA and RNA, but we work with the other 4 fields, and I will give you some examples on how we can utilize and what is click chemistry, but also how can we utilize it within these areas. So my inspiration really comes from this forest in my grandpa's farm, where I wanted to understand what is the communication between the plants that make them understand what is a neighbor, where the insects getting their knowledge of where to go from, and so on. And all of that is an interaction of chemical compounds with receptors and other things. I didn't know at the time, but I got a real interest in understanding it. And that's the starting point. And this is the next step, making the gunpowder. I made the most beautiful rockets you have ever seen. And I could go down to the beach and fire like 5 of them, and then I had to rush home because the police would come. And that is because they actually went up to 300 meters high. Okay, in young people, when they come to me and ask for a position, the first thing I do is ask for their soft skills. I think today factual knowledge is not that important anymore because we have AI, we have the internet, we have so many different things that can help us with the factual knowledge. But what is very important is that you have intuition, that you are able to imagine things in your brain, that you have courage to tell your PIs, no, I don't want to do what you tell me, I want to do it my way. And this is a very important property of young students.
So sorry all PIs in the room, but I think you will have problems with your best students mostly. Then creativity, ability to observe and use the observations creatively to design new things, to make an observation, a new phenomenon, and so on. But your road to the successes is not going to be straight. It's going to be a very bumpy road because success doesn't come easily. All projects that are designed behind an office desk has a tendency to fail. So your PI sits and makes a very good suggestion, looks good on paper. You go to the lab, it just doesn't work. 1 month, it didn't work. 3 months, still didn't work. Then you have to make decisions on what to do then. And you rise again and you go outside your own comfort zone into other fields to find the information that you need to solve your problems. You are persistent and you remember to study all your life if you want to be a scientist. So this here is T-Rex. Let's call him Donald T-Rex. Donald T-Rex is controlled by a very simple molecule, the aggressor molecules here, and his brain here in the brain is very, very small compared to the size of his being. He is also controlled by pleasure molecules like dopamine. I can't do that. That's really irritating. Like dopamine or serotonin. Dopamine is a pleasure molecule that makes us do what we did again. And serotonin is our guiding molecule that is the cognitive processes that is controlled by that. I think Trump is mainly by the 3 phenylalanine derivatives there. Then we also have this molecule, which is obviously absent in Donald T-Rex. But this is a molecule that's needed if we want to have the advantage of being a herd community. As a herd community, we help each other, and that is actually the key to the success of the human being. We are a herd community where everybody has a place, and we should help each other. This is not what is happening in the world right now, as you probably know. So this molecule has a very low value today. It's present in all human beings. Of course, it's more present during lactation in the woman, but it's actually present in all of us. And what it does is that it binds to the receptor here. It looks like that actually in 3 dimensions, and that's what the young people should understand. What does it actually look like in 3 dimensions? And then it binds up there, so you have complementary interaction of blue with red and red with blue, and then you get a reaction through the receptor into the inside. You get expression of proteins, and you feel empathy, and so on. Then you can see nature is cheap because it's just taking the love molecule and make a blood pressure molecule, also regulating something in the heart, by just modifying 2 residues here. We were very interested in this kind of chemistry to understand what is going on with peptides, so we worked with peptide technologies for my whole career, and this is some of the areas that we've entered into using peptides. We also worked on quantitative chemical transformations up to the 90s because of the combinatorial era of chemistry. We worked with transformations in chemistry that could be used combinatorially. And that means that we need quantitative reactions. Today, you might call some of these Klik reactions, and that is actually what happened during this period. This is Carlsberg Laboratory, a wonderful research institute at the time, and I got funding from Danish National Research Foundation to follow up on this quantitative chemical reactions on solid support. So we developed a polymer which was highly polar and swellable in water, where enzymes could migrate into the interior and interact with the molecules in there. So this is, in this case, if we make a combinatorial library, one experiment in each of these small beads. We also built a platform, and here you see the combinatorial synthesis, 1 week to make 500,000 compounds. Then we screen with enzymes, and you can see some blue beads here. We pick those out, and then we understand what the enzyme is doing when we analyze the structures of these molecules on the beads. We can resynthesize, and here we have a mouse that, where we have injected one of these into the tail vein, and it has arthritis in this foot. So now we can understand when a certain enzyme is active during an arthritis disease, and can we treat that. We also make these kind of molecules I talk a little bit about. We make catalyst, and this here shows you cells that are grown as a monolayer on the surface of the beads, acting as a detector for interaction, and you see here an active bead in the presence of inactives. We isolate that, analyze what is on it, so we get receptor interaction, and we can do that also by encoding. This is like a small barcode that we have in each of the beads. We have immobilized fluorescent microparticles. They make a matrix, and this matrix is our barcode that we can read. So this is our platform technology, developed partly in Carlsberg and partly at Carlsberg Laboratory. Yes, please. So we were working in this time with making difficult peptides, and we were using acetoacid chlorides for this, because they are very reactive amino acid derivatives with a very small protecting group. So we could make peptides that you cannot make in any other way, like this one here. And also we thought that we can use this reaction maybe to react with other nucleophiles. So if we use this ACE chloride, maybe we could take a C nucleophile and make this compound. That would be very useful, because if we include that in a hormone synthesis, we can put a lipophilic chain on with a Diels-Alder reaction, and boom, we have one of the slimming reagents that are used today. But we didn't do that because it didn't happen. We just could not make that work. So Christian Tornø, who was working with me as a PhD student at the time, he tried and tried and tried, and he came to my office and said, maybe, Professor Meldal, this is not the right thing for me. So I think, can you help me get a job at Novo Nordisk? I said, no, maybe we should just look at it. And we looked at it, and we found the proton, which is crucial here, and that's the proton of this triazole sitting there at the bottom, this proton here. We had formed a new product, which was not really expected at all, and it's when you see these unexpected things that you have the potential for making something really new. So we compared this reaction with all the different biomolecules, like amino acids and carbohydrates and so on, to see what is the compatibility. And in 2001, Christian went away to California and presented his work with the triazoles here, together with one other of my PhD students. So Christian was very proud. I here have a drawing that shows what an alkyne looks like. It's a sack of electrons. So this is the HOMO of the alkyne. And that is looking out for electron density. So here, copper 1 atom is sitting on the alkyne. For some reason, copper 1 has a huge affinity for alkynes. So it sits on the electrons and extracts electron density. It becomes acidic, and the copper takes the place. So that's the first step of the Klik reaction. And now we have a second copper atom coming in here, and it is also binding to the electron density, and it's stripping off the electrons like that from the alkyne, opening up the electrophilic center there, and then it has a LUMO looking like that. So from the side, we get now an A site coming in with the full electron. So these are the homos of the LUMO on the below and the HOMO on the top, and they make the two new bonds. If we look here, this is what it looks like. A metallocycle that loses first one copper atom and then a second copper atom. And the driver here is the very high enthalpy of reaction when you get an aryl group. So Christian had now connected two small molecules with each other, and that's not very exciting. But I'll show you how this can be used. One caveat with the Klik reaction is this oxidation of copper 1 to copper 2, very low energy barrier for this oxidation. The problem can be solved by adding something that reduces it back to copper 1, but that is not good because in this process we form a lot of radicals that reacts, in particular if we have a biomolecule like a protein, and it oxidizes a lot of different places in the protein, and that is what you see down here below. We see plus 16, plus 40, 32, 48, and so on. So we can completely destroy our molecule if we don't remove the oxygen. Next. So we do this under protective air, and this is just standard instrumentation for our cyclizations, where we use an alkyne and an azide to cyclize a macromolecule. It works very well by a slow injection of the peptide into the mixture of the Klik conditions in here, and then we get the product formed in good yield. It's not so easy to make a Klik reaction because you have to fulfill a lot of different requirements in order to have proper Klik reactions. And that's why we only have the TCO reaction, the QAC reaction, the spark reaction, and maybe some nucleophilic aromatic substitution reactions today as Klik reactions. It's not something you find every day. It would be good to have a lot more, and I really encourage the young people to look out for reactions that have these kind of properties. So if you install a Klik moiety in a protein or peptide, like shown here, we can do that by amber stop codon technology, where we have insertion by biological means of these residues into a protein, so unnatural amino acids in a protein, or we can synthesize them with these moieties in there. And now we mix those with copper 1, and we steer them overnight, and in the next morning, we can come and collect this molecule very cleanly. And now we can use that molecule to recognize a cancer cell, to import it into a cancer cell, and to inhibit the inhibitor of programmed cell death. So what will happen is that the cell decides to undergo programmed cell death. This is something that is in the works right now, actually. But there are other ways that Klik chemistry has been utilized in cancer research quite successfully for very selective delivery. We can make these small molecular robots, which are multifunctional molecules. So we have 3 functions in one molecule. That's a small robot. Or we can make ligations, we can make stabling of structure, and so on. There's so many different ways you can utilize the Klik chemistry because it's a completely orthogonal chemistry to all the other chemistries we have. It doesn't interact with any of the other chemistries, and any of the other chemistries don't interact with the Klik residues. So these guys here are 400 million years old, not these ones, but the species. And it has a very unique innate immune system where you have 2 macromolecules that comes around a bacteria that infects this, and then it injects toxins that kills the bacteria. This is one of the toxins up here in the corner called targeplacine. And we were interested in targeplacine because of the structure. The structure is a beta hairpin with 2 disulfide bonds. And what we wanted to see is that can we keep the biological activity when we substitute these disulfides with triazoles? Because if we can bridge with triazoles, we can make completely stable compounds. And we did that. This is work from Kai Holland-Nell. And here you see a cell membrane. And when you then react that with the compounds, it forms a pore, and everything from inside the cell flows out, and the cell will die. So this is a very good antibiotic. We tried it with 4 different species, and we tried the natural targoplasine and our molecules. And here you see the result. Our molecules, which are the 2, the green and the red strip there, were totally active all over the place. The natural one was active for the 3 first one, but for Bacillus, there was no activity whatsoever. Probably Bacillus subtilis has this ability to reduce the disulfide bonds. So we had here maybe a good way to do antibiotic resistance, convert that into an active antibiotic. Here we are. This is the first day of the holiday that I recommend you all to have in Stockholm. You were invited by the Nobel Committee. And this is the day where you go to the museum and you are told about what is going to happen during the week. And here is one very important thing that you deliver. It's now in the museum, together with a publication that describes it. It's a small multi-column synthesizer that we can use also for combinatorial chemistry to make millions of compounds. So this was really important for my career. And this was actually made in the garage in the neighbor's house. He had a slate, so I could actually carve out in Teflon, and that was really cool. So you also have to sign a chair, and here's my signature, side to side with Benjamin List and Carpentier. So I'm very proud of that signature there. Probably the best signature ever. Next slide, please. Molecular recognition. Every single thing that happens inside of you is preceded by an event of molecular recognition. Everything is specific, and that is based on 2 molecules coming together, recognizing each other. Here we have a process that we have developed which simulates the main recognition molecule in the world, which is the antibody. Everybody knows about antibodies. They are really good in recognition, down to picomolar activity. These are what we call beta bodies. They have a structural site that we maintain all the time with threonines, beta-branched amino acids on the top side. Then it has a bridge and a turn, and then we can vary 10 amino acid residues to recognize any surface. And the good thing here is that we can design that for any surface in a protein. So we can select a position on a protein and make one of these molecules. Here we actually made 2 molecules, the red and blue ones. They are both the first attempt to design recognition molecules towards this protein. And we went to the lab, synthesized them. When we mix this one, which is sitting on the bead, with this one, which is fluorescent labeled, nothing happens. Even after 4 hours, nothing has happened. And then we add just 20 nanomolar of the green and orange here in the middle. So that binds to the one on the bead, and then it recruits the fluorophore on the backside. So we have a very good sandwich assay made entirely on the computer. So that is a really strong technology. Can we use that for anything? Here's an Alzheimer plaque in a brain of a dementia patient. And Greenman Allied, they isolated the fibrils here from the plaque and did EM, so electron microscopy, to find a structure that's actually present in plaques. And then we cut that in 2 to have the 2 growth phases, one on the top and one on the bottom. They are different. And then we designed molecules that sit and bind to that in order to prevent defibrillation. Next slide. So we designed 2, one for each end of the fibril. And when we added these, so this is the fibrillation curves here, where we insert this into the beta sheet structure here, and we get fluorescence increase. That works all the time here. And then when we add our molecules, you can see we get a little bit of fibrillation initially, and then it disappears again. We can also dissolve formed fibrils with these molecules. So that's very promising. We were not quite happy with this first part here, so Yan Sang in my laboratory designed a building block that we could use for solid phase synthesis based on glucosaminic acid. So this building block has the same stereochemistry as D-threonine that we use for the structural part of the molecule. So we could just incorporate those instead of the threonines. And what happens now is they stick out hydroxyl group arms with the same number of hydroxyl groups as a sugar, so a glycopeptide, and they interact with the molecules in the water and make a hydrogen bonding network across it, so the molecule becomes much larger and much more rigid. And next slide. Next. We incorporated them several places in these structures, and we synthesized 5 compounds. Next. Shown here. And we tested those for the fibrillation process. Next slide. In different combinations. And you will see for a beta 1-42, the long peptide from Alzheimer, we see absolutely no fibrillation when we combine these 2 molecules with the fibril-forming molecule in a ratio of 1-to-1. Here you see the fibrillation and different combinations, and this one is completely inhibiting the fibrillation, the same for 1-to-40. So we have a very efficient system now to prevent fibrillation. And this is what it looks like. So here you have the water shell, hydrogen bonding network interacting with the hydroxyl amino acids, and that creates this kind of molecular structure, quite large, rigid structure that fits exactly onto the growth phase and helps the dissolution of the fibril. Next slide. So we imagine that we get these fragments of fibrils that we hope to demonstrate we can wash out during night to clean out the brain eventually. That would be really cool. During this program, we also studied possibilities to cleave the Alzheimer peptide to avoid the fibrillation as a procedure for avoiding that. And we, next. And we made metallopeptides containing carbines that could bind to the transition metals in order to get hydrolytic peptides. So these should be catalysts that can catalyze that. We combined them with a lot of metals, and it worked fantastically. So all of these combinations cleaved the Alzheimer peptides like that. Great. But the fact was that so it was so successful that my PhD student decided to make the control and take out everything except for the Alzheimer peptide itself, and then it still worked. So here's another moment of observation where you don't throw your things in the sink, but you actually think, is here a new thing, a new observation that we can go with? If you go to the next slide. We found that this is an autocatalytic process of the Alzheimer peptide. So maybe even something that is important for systems biology. The first proteases in life could be uncoded proteases that are simply peptides coming together forming proteolytic aggregates. This is a proteolytic aggregate that forms slowly. If you go to the next slide, this is what we synthesized, a lot of peptides to investigate that, and then we tried them out. If you look at the red line here, this is a starting point. After 11 days, and this is important, there's a lot of lag time here. After 11 days, we see the formation of a second compound, then a third compound, then this one, and then this one. And we have all the mass spectrometry to prove that. And it's right here in the nucleation site that this protein naturally have a tendency to cleave itself. So that's very exciting. We could take a mixture here of these compounds and add to the 1 to 28 peptide. And you can see after a latency period here, we start getting the hydrolysis, and we have complete hydrolysis in a short period of time. We can see it here for 1 to 40, adding a little bit of that one, just 1 microliter to a milliliter, we get this result in 12 days. So we are forming an active aggregate that is self-promoting. So it starts forming itself as soon as it starts hydrolyzing. Can you go back one? We also looked at this for the long one, the one that really is difficult to dissolve because it forms fibrils. And that takes a long time, which is good and bad, because that means that it's almost completely precipitated in the brain. And then eventually a little bit of it gets into solution and becomes proteolytically active, and that's what we see here. My student put this on in May last year, came back in June, nothing happened. July, nothing happened. August, nothing had happened. And then on the 15th of August, it looked like that, suddenly. Long latency period, and then it comes autocatalytically. We also found that this process is initiated by formation, the fibrillation is initiated by formation of these donuts, single-stranded structures like that collapse into small sticks. We see that all over the place here, small sticks that are formed, and then these sticks come together, form a long train. Here, sticks coming together, forming a long train, which maturate and give you the stable fibril. So this is why this is important. That is because it could correlate to the neuronal cell death that you are observing in Alzheimer. In neurons, in the presence of Alzheimer peptides, you have cleavage of this important molecule called NCAM2. It's a molecule that is a survival molecule of neurons that keeps them together, allow them to communicate and form connections and so on. So this is a very important molecule on the neurons. And if we look at a control, we see very little of this cleavage product. If we look at the Alzheimer patient, you can see a lot of this soluble peptides formed, and in particular in the vicinity of, if you take the next one, yeah, this is what it looks like in the membrane. And if you take the next one, in particular in the vicinity of Alzheimer's, so here you have an overlay of the soluble NCAM with the Alzheimer peptide, and you can clearly see that it is in the same region. When you look at the cleavage site of the NCAM, it also looks impressively close to the sequence of the beta. You have 2 phenylalanines here. Here you have 2 hydrophobic isoleucines. So there's a lysine that's charged down in this end. So it could really be that this Alzheimer is the reason that we have neuronal cell death, which would be quite exciting. We don't know yet, but we are investigating it. So here's what it does. We have the NCAM molecule, and our aggregate comes by and recognizes the sequence sitting down there, anchoring it right into the membrane, and it cleaves off, and that, of course, is detrimental to the neuronal cell. So we have the formation of the fibrils, the mechanism. We have the formation of the autocatalytic process here, and we have molecules that can inhibit the fibrillation. So I think we have very good tools in the Alzheimer field. Now we are back in Stockholm. This is the day where you give your big lecture, and you're very nervous because you're talking to 10 million people. But here it is. You pull yourself together, you get it done, and after the lecture, everybody is very happy that it went so well. So this is Barry and Carolyn and me. Yeah. The TAT peptide is nature's ultimate cell-importing peptides, and cell-penetrating peptides, or peptides that can import other molecules into cells, is extremely important in medicinal chemistry for the pharmaceutical industry, because most of the targets that we want to get to are either in the brain or inside the cells, not on the outside. Maybe 20% is on the outside, the rest is all hidden away. So we need molecules that can penetrate the cell membrane. A huge barrier in biology is the cell membrane. Penetrate that and get into the cells. So this is the natural one, the TAT peptide from HIV, very efficient. It penetrates with this part and it recognizes molecules to transport with this part. We made a small analog to that and we wanted to make it less basic. This one has a lot of basic residues and that's quite toxic for cells. So we want to make something less toxic and we made this small construct. We made actually 16 of them and here you see a Klik bridge. You see the positive charges, you see hydrophobic material here, and we were successful, as you can see here, to get really good import. I would like to show you the chemistry. So we start by forming a protected hydroxyproline to make this molecule here. We have to have a derivatized proline. So we make a protected hydroxyproline, we alkylate it, and we reprotect it first by removing here, then protecting here with FMOC, and we have a building block. We can do the same up here by substituting first with A site in a double inversion. So we transform this into a leaving group and substitute with A site. We reduce the A site. We then do the reductive amination. We add a Boc group, and then we reprotect it with the FMOC to get a building block. That works very well. And now we have two building blocks that we can use combinatorially. So here we have them, 4 building blocks that we can use combinatorially. And we synthesized 16 peptides like that and purified them and tested them for cell penetration. And here you see the result. It's quite difficult to see from where you are, but really nothing happens. It's very boring in all of these, and then suddenly down here you see one which shows a lot of import. So 1 out of the 16 were really, even though they are so similar in structure, 1 out of the 16 were really, really efficient. Next. And this is a quantitative efficiency. We can synthesize these. Here are the mass spectrometer and the mass spectrum and the chromatogram. Synthesize this and this one, which is a TAT peptide, and we can compare the two. So this is our synthesizer. We build a lot of those for the students in the lab, and we can synthesize very efficiently on those. We investigated the mechanism, and here you see the mechanism, and it's temperature dependent, so it's an energy requiring, ATP requiring mechanism. We can see import already from 100 nanomolar concentration, and the import is at its maximum at 12 hours. Next. We also investigated how it's actually occurring, so we added different inhibitors for the different pathways, and we found that this is a dynamin-dependent endocytotic pathway, so we have an endosome formed, which is a dynamin-dependent process. We could also use this in all kinds of cell lines, so that's very promising. And next slide. We could even take this molecule, the best one, and supply it with an alkyne, and then we could link it up with green fluorescent protein. So we have this one, and now you can, next one. And now you can see with just GFP, nothing comes in. With GFP plus this in solution, nothing comes in. But with the connected ones, go back. With the connected ones, you can see very clear import into the cells, both with MCF7 cells and HeLa cells. Next slide. So we think that we have this molecule inserting into the membrane, so it's not actually transporting across the membrane directly, but it's inserting into the membrane, making this vacuole that is then imported into the cell. And depending on then what we have on the arginines here, we can either have contained in the endosome or we can have a release from the endosome. We have made it with 16 butyl groups on the arginines and that's very efficient for delivery intracellularly. This one forms these endosomes, which can then be used for transport across the blood-brain barrier. And this is what we are investigating at the moment. So a very powerful technology. Next slide, please. And here we are back at Stockholm. This is the day where the king come up, and you are approaching him. He gives you this one. And you're very happy. You say thank you to the king, thank the public, and you thank the committee, and then you go back to your chair. On the way there, somebody comes and pulls it away from you, and you're thinking, did I say something to the king I shouldn't have said? But that's not the case. The reason is a lot of us made our discoveries 30, 40 years ago, and some of us have gone a little off in the meantime by Alzheimer or other ways. So a lot of the laureates actually originally forgot their medals in the ceremonial hall. So now you get it on the last day, and then it's your family who makes you take it home safely. Okay, next slide. So this here is a protease tab that we try to express in coli. The coli are not very happy because when you have a protease in a cell, it can cleave the interior proteins, and it doesn't like that. So we can get very little initially, and then it dies off, and you don't get any protease product in expression. So we thought maybe we can just cleave this in two. Next slide. Here. Yes. No, that was okay. Cleave it in two up there, and express these two parts separately. They wouldn't have any proteolytic activity. And then include some Klik moiety to regain the connectivity. And we did that. Next slide. And the coli now are very, very happy, and they produce large amounts of the two fragments. And now we can take the two fragments. First we asked the coli, is this okay? And it was very, very content, because it gave us a lot of molecule. And then we could combine the two. Next slide. We have combined the two here as two halves that comes together and form either a long bridge or a short bridge. And we compared those. If you look at the next here, here is a synthesis of each of these parts of the molecule. One of them is here with the mass, so you can see it's very efficient. And then when we mix two of these and click them, with a short bridge, you get some clicked and some unclicked. And with a long bridge, you get a complete conversion. 1-to-1, you mix them, and you get a new protein. So that shows really the potential and the power of a Klik reaction with a completely unprotected protein. We tested these two fragments that we have made and clicked together. And next. And they come here, and then a copper ion comes in from the side. Oops, just stay there. Comes in from the side, and click them together. You get the triazole here. And this then, don't touch anything. Then we have it there. And the reaction that we are investigating is the cleavage of this substrate. And our protease is actually a little bit better than the natural one. So, and this is probably a way to go if you want to make large-scale expression of proteolytic enzymes. Another way could be to make an inhibited version of the enzyme and then cleave off the inhibitor after the production. But here you can produce directly the active protease parts. Next. We're back in Stockholm, and this is actually the last day, the last picture from Stockholm. This is a ceremonial dinner, and on the first day in the museum, my wife had spotted the Klik dress. There's 6 mannequins with dresses for the Nobel Prizes. Klik dress was there in the middle, and she looked, that looks nice. So she called the Arts and Science Department of Stockholm University and said, can you also make me one of those? And they said, yes, we can do that, because that would be a win-win to present this dress for the king. And this is my wife, and you can see how beautiful she is dressed there. There's a real danger when you dress your wife like that. And in this case, the danger is called Prince Philip of Sweden. He spotted my wife, and I was sort of guided over to the side, and she was going with Prince Philip to be seated between him and Prince Daniel, and I was down by the door in the draft. That was okay, because the next day we were invited to the castle to have a dinner with the Queen, the coming Queen of Sweden. I would like to end with another kind of Klik reaction, which is the Enig reaction. The Enig reaction was discovered also by serendipity, like any good research. So we discovered that when we try to make N-terminal peptide aldehydes and do reactions, all sorts of electrophilic, nucleophilic reactions on the N-terminal here, we had a problem because this one would react with that, and then it would react with other things from there. So when we make an aldehyde here that is protected and deprotected, we form the reactive aldehyde. It reacts with an amide bond, and that is completely unexpected because amides are unreactive, essentially. So it reacts there and gives us a hydroxylactam that forms an N-acylaminium ion, one of the more reactive species that we can find. And this is reacting with C nucleophile. In this case, we have a tryptophan, so we generate this heterocycle. And that's a very nice reaction because now we can transform peptides into small drug-like molecules. We first synthesized a lot of building blocks here by a double malonic ester synthesis, introducing the aldehyde first and then the side chain. We could also do an acetic ester synthesis here to form this kind of molecule. And then we could do oxidation of diols to form first a carboxylic acid and then an aldehyde that we protect. And we can also do oxidation of C double bond to get aldehydes at any time. And then we can reduce the amino acids on the acid side to produce an aldehyde and then couple it via the amine. So this is how we generate aldehydes from alkenes. And this is how we generate an aldehyde protected here on this side, and then a reactive end here as a urea. So we have now a lot of opportunities to form these reactions. And here you can see 4 examples of these reactions and the crude results that we get out of that. Complete control of stereochemistry, 100% conversion into only one product. That sounds like a Klik reaction, right? Next. And if you look at the NMR, again, crude products. So students, this is what it should look like when you take a crude product, go to the NMR, put it in there. And if you have this, you are in good shape. Next slide. So we could even use unreactive C nucleophile. Next slide. Here we have a dichlorophenyl group, very electron poor nucleophile, and it still forms quantitatively this product. So that's quite unusual. It's because of the entropy-driven reaction here. Entropy-driven reaction here, and the fact that you have a very reactive N-acylaminium ion. And it goes 100% stereoselectively. Next. We can also use NO and S nucleophiles in very good yields of the products. Next slide. Getting you these kind of heterocycles, also very interesting from a pharmaceutical point of view. And if we don't have a reactive side chain like here, it will just find the next amide and form this kind of optically active compound. So we can use this also inside peptide chemistry. So here we have the mu receptor agonist, the opioid mu receptor agonist, and it is formed from making an aldehyde on this double bond, and it can go together here. So here we have introduction of allyl glycine. We do the aldehyde here, form this heterocycle, and then we can continue the synthesis. Or we can complete the synthesis and then carry this reaction out at the end. That's a very good reaction. We can also use these amino acid derivatives that I talked about and make this kind of heterocycle. So we now have this activity and this aldehyde, and we can get this kind of product. These are very interesting because they do the oxidation here on the center, and we get this compound with air, or we can use DDQ, and we get very highly fluorescent compounds. So now we can make peptides that are in situ fluorescent. So we have a very nice combinatorial template. We can make 5, 6, and 7-membered rings. We can make 5, 6-membered rings on the other side. We can use C, S, and N nucleophiles. So there's a whole lot of variety we can do here, and that is just the ones that form the N-acylaminium ions. We also have the carboxylaminium ions forming a whole new set of templates. So all in all, next slide, we have made quite a lot of different structures, and all of those are like a goodie bag for pharmaceutical industry because they are out of flatland, and we can put any kind of residues around these templates. So to end up, I would like to say the world is an increasingly dangerous place to be in. More and more conflicts, more and more problems with global warming. I'm very sorry for you young guys, because we left you with a world that is in big trouble. And we have to solve that, and we have to solve it together. It's all our responsibility, in particular us as researchers, to have an attitude towards getting this problem solved for the future. And the problem is that we have an autocratic trend, because when you start seeing problems, human beings have a trend to back up behind a strong character who promised to solve all the problems, but never will. That is just postponing the pain. We really need to take this seriously and get a solution to the problems that we are facing. Because this is ice melting, maybe turning the Gulf Stream around, getting an ice age in Northern Europe. It's a desert belt broadening. It's flooding. It's burning of a lot of important cultural heritage in wildfires. There's so many things that goes on at the moment, and we have to stop it. So education is the solution. Not only of you as experts in chemistry, you will probably be world-known chemists in a short while, but that's not the most important thing. The most important thing is that we also inform the general public. That is why I think it would be good to have education of the young from the first grades with an animated three-dimensional world of chemistry. So here's my suggestion. We make this repository of videos and VRs, and you call the AI. Yes, you call it, and you get the help, which is for that week or that year of the students. You get VRs and videos and so on, and this can easily, next slide, so you get the videos and this can easily be translated and globalized. So if we can make this together, we can make a really strong case for the future, not only for you as specialists, but for the broader general public. And this would be important for our environment, as I said, but also for climate, for the global politics, conflicts, maybe even religious conflicts, and we can have different points of how our resources are actually utilized, wealth and peace in the world. So an important, I don't know if that's going to work, but I think it will. And this is something I gave to Lego in Denmark. You know all Lego. This is a molecular system that actually behaves like a real molecule. So I think we should have these kind of building. We can make it. I made a patent claim for Lego to make this, but they don't want to produce it, and the reason is they are not square blocks. Okay, but they behave like molecules, so there's a small resistance to, when you turn around the sigma bonds. There's magnets for hydrogen bonds and all of that. And I think it's important that we introduce these kind of tools to get to the children. This is a child just born, have no language, only learn by vision. We have to utilize that property early on to get an instinctive understanding of chemistry. Next. So what we really want is productivity. And it's important that we have two kinds of research. So today, most of it is in one big bag, and either you do applied or you do fundamental research. But these are actually two completely different disciplines. Taking research already existing and transforming it into application for society is extremely important, but it's just as important to secure the day of tomorrow with new fundamental research. So that is why we should split, politicians should decide to split this up into two entirely different regions, areas. Look at it this way. And then give us the money as fundamental researchers as well. Give us it not based on what we will do, but on what we have done. So of course you have an entry into the system for first users, but once you are in the system of research funding, you just need to present what you did the last 3 years. That is extremely good. You get extreme amount of money. If it's mediocre, you get mediocre money. If it's nothing, you get nothing. That would very rapidly clean out the research system to get the best results ever from our research. And education, very, very important. Here is Finland, South Korea, Poland, and Vietnam, all countries that elevated the society by doing a deliberate investment in education. Different ways to do it, but they all succeeded extremely well in elevating the society. So I think that you young people should consider where can you put your efforts to generate a secure, sustainable future. And here are some of the areas that are very important. Energy, of course. How can we utilize? We have plenty of energies from the sun. Why should we utilize the fossil fuels that has been stored in the ground for 4 billion years? Why should we utilize that in the breath of a generation or two? It's stupid. It's plainly stupid. And we have so much energy from the sun, we just have to agree on how to utilize it and not bomb each other to death. We have here a vehicle for transport. This is not the one I have in mind, but I think we should look at our transport. It's highly inefficient as it is today. A lot of money could go into making transport that's much more efficient, underground and individual still. So for all of that, we need excellence. You are the excellence of the future, not you individually, but the groups you will be working in. So if you are an excellent person, you can have an excellent group. All of your colleagues are part of that excellence. So we need excellence in playgrounds that are well equipped. That's why we need infrastructure for your research. Very important point. And you are allowed to do what you want to do. Even as a young researcher, the PIs should be told not to be too decisive about what to do in the research programs. Because that allows for you as young students to come with your ideas, to formulate your own approach to the problem and solve it. As I said, usually the first office-mate project is never successful. And then I would like to thank you for your attention. I'd like to thank my collaborators. I would like to thank the people from the university and the people from Carlsberg Laboratory. And most of all, I would like to thank my family for being very patient with me and also to have scientific discussions because my wife is a scientist and we have very good discussions. She is keeping me on track most of the time. So that's a really nice thing to have with your partner. Thank you very much.
Thank you, Professor. I'm here and we will have a seat here for the questions and answers part.
Yes.
So there is a lovely audience, and so I would like to introduce all of them, but first I will give a kind of the Turkish comment first, and then we are going to sit here together.
Yes.
Yeah. Soru ve cevap kısmına geçiyoruz. Lütfen soru sormaktan çekinmeyin. Türkçe sorabilirsiniz, biz çevireceğiz. Dolayısıyla da sorularınızı bekliyoruz. Arkadaşlarım destek olacaklar size. So it was a really nice talk. Thank you for all efforts. So sorry about the technical problems.
Probably that has to do with my
Yeah, there
remote
could be a kind of
because when I took it out, it worked okay.
Yeah, yes, we can have it.
Do you want to keep something behind?
No, you can just take it off.
I think you'll put it.
Yeah.
Would you like to have some water?
Don't say.
And just close it here.
Arka planı başka bir şey.
Yes, come on, please. Okay. I think you can also hear the audience excitement.
Yes, yes.
Yeah.
And we already have a question here,
Yeah.
which is very nice.
There will be questions a lot, so we received some of them, but first I would like to introduce the audience, because
Yes.
there are different representatives of the society here to listen you. Gebze Technical University is one of the research university and really open to open science activities, especially for scientific communication. We believe a lot. Last year, we influenced all Turkish universities to install a scientific communication offices by the help of the Higher Education Council, and so their representatives are here also. And so we have high school students from different, high schools.
Yeah,
so
I
they are
see.
really talented students and loves the, research part a lot. And our lovely students and also faculty staff and from different universities that we have the guests today. And so different NGOs and representatives are also here. So it's a great something, and so I would like to have some questions first because of the priority thing here.
You
Sorry
have
about
priority.
And so, across the world, scientific communication is quite important to deliver all these things, and your last slides were really inspiring for all of us, because it's a kind of, you are doing lots of things, and so your all talk was related to chemistry, but also biological sciences. So I'm really happy to have also cell pictures there, Western blots, and also, they are all connected to each other, like the agriculture to Alzheimer, and also for other disciplines, and also engineering part. So this is really important, I think, and in this context, the Nobel Prize is not only a recognition of scientific, excellence, it's all kind of the powerful bridge between the society and science. So may I ask, how has receiving the Nobel Prize changed your perspective on science communication and your role in engaging with society?
Of course, the communication becomes much broader when you get a Nobel Prize. I used to have a very strict rule about having new things to present on every single conference, having all the experimental in all my papers, only present things that were published, and so on. All of these rules are gone, of course, when you get a more broad presentation or communication schedule. So now I'm communicating very much also outside the actual science.
Yeah.
Yeah.
So,
And also I have a lot in Denmark. I have contact with schools and also non-university education institutions, which are educating grown-ups in different fields. I do that a lot.
it's great ambition and it's a great passion for chemistry and also for science, such as our Science Communication Office every year, welcoming to over 14,000 students in this campus, because it's a really green and lovely one of the campuses in Turkey. And so for this purpose, we believe a lot, any person can change the world with small steps, and so do some really critical things. I little bit read about your previous podcast and also things, so it's really nice to see you like playing guitar.
Yes, I think music is an alternative way of using your brain to the scientific process, which is more, here we have a challenge and then we are going to a pathway in our brains to get to some sort of solution over here. This is a very long and tedious process. It can take hours to go from A to Z. And in music it's completely different, because in music you are right at the moment, and particularly when you are improvising, you need to figure out where to go right now with the music that is playing behind you. So I think that is a very nice alternative, very relaxing actually, compared to the scientific thought, which can be quite headaching sometimes.
Yeah, it's a great hobby also.
Yeah.
Yeah, or also I learned about that you like the Lord of the Rings a lot, I think.
Yes, I think that I love this kind of fiction that you have in Lord of the Rings, or
And as you can see,
The Matrix. Wonderful movie. Wonderful movie.
yeah, it is. Such as I got some quotes for the Lord of the Rings, which could be really reliable for this event. Galadriel is saying that even the smallest person can change the course of the future. So maybe small steps from all over, the world is going to be really a bit full of empathy also.
Yeah. And we should remember it took Tolkien 10 years to actually make, or maybe even more, to actually make this beautiful world that he is describing in his book.
Maybe connecting more,
Yeah.
or talking more,
Yeah.
or maybe supporting each other.
Yes, and be sure that we do the right thing rather than the quick thing.
Yeah, it's a kind of the scientific also ambition, I think, the connecting more. And
Yeah, but there's this huge pressure for you as young people to actually come up with publications all the time. And it's more important that you have thought it through than that you publish your paper. So I really encourage you to look at that, because a lot of the literature today, a lot of it is actually useless if you try to repeat it. And this is a problem for science. Science has to be scientific in the sense that when you do it again, it's happening in the same way.
Yeah. And there will be questions from the audience, and so may I have kindly see the microphone carriers? Yes.
I think
there
the
is
first one, very, very, yes.
Çevirirseniz sevinirim.
Tamam.
Hocam hoş geldiniz.
Welcome.
Aziz Sancar hakkında görüşlerinizi söyler misiniz hocam?
Tam anlayamadım ben.
Can you translate that?
Yeah, I will.
Aziz Sancar hakkında görüşlerini söyler misiniz hocam?
Tabii. So he's saying that warmly welcome, and also wants to know your opinion about the Aziz Sancar. Is one of the Turkish Nobel laureate, received the Nobel Prize for the biological clock. He's asking your opinion.
Yeah, so the
about
biological clock is extremely important, and he truly deserved that. I think that mostly, if you look back over time, the Nobel Committee has done an amazing job in finding exactly the right people for the Nobel Prize. Because what they have done has always been shown later on to have extreme importance for humanity, which is one of the criteria for giving the Nobel Prize. And the biological clock, understanding how it is influencing all the biological processes, is of course very important. Our aim, a lot of researchers are aiming at getting longevity in life, a healthier life towards the end, and so on. And there the biological clock is extremely important as well.
Of course. I'm going to translate your answer. Biyolojik saat konusunda Aziz Hoca çok önemli bir ödül almıştı ve aynı zamanda şu anda uzun ve iyi yaşamın ne kadar araştırılması önemliyse biyolojik saat de aslında o kadar önemli bir buluş olarak önümüze çıkıyor ve Nobel Komitesi'nin belki de en iyi organizasyonu bu noktada gerçekten ödül alacakları en iyi şekliyle seçmesinden ötürü. Dolayısıyla başarılarını kutluyoruz şeklinde alabiliriz. We can have another question.
Yes.
There is a young lady there. I will return back to here, don't worry.
Hi, I really enjoyed your talk, and as a high school student learning chemistry, I was wondering whether Klik chemistry emphasis on simplicity and reliability could influence how chemistry is taught to students like me.
Yeah, so I don't know that you shouldn't learn the thing that we have learned as young people, because chemistry is a real scientific discipline, meaning that it's always correct. And you will have to learn. I have a course in the University of Copenhagen, which is called the Meldal kill course. So it's supposed to kill the students, but it doesn't actually. They get very challenged, but they also get very happy with that course because it's really concerned with all this knowledge that we have about organic chemistry, how the electrons are moving around and so on. And Klik chemistry is just part of that. The reason that Klik chemistry becomes so important is because you can suddenly do reactions and architectures, molecular things that you could not do before because there was simply not the tools to do it. So that's where Klik chemistry has had great importance. And I think you will see this reflected in new ways of doing medicine and so on. So there, of course, students will have to learn things differently.
Yeah. Yes, we can have another question from here, and then we will return to this side.
Thanks for this nice talk. Professor Morten, so last year's Nobel Prize was based on the reticular chemistry in the field of MOVs and COVs, this kind of structures. And also the Nobel Prize winners mentioned the importance of AI, artificial intelligence, self-autonomous labs. So I'm actually wondering your thoughts about how, for example, Klik chemistry can be used for programmable, for example, this kind of structures, or how this Klik chemistry can help us to, transfer to the self-autonomous labs.
So AI is mainly a machine that learns from a lot of data. We don't have this huge amount of data on the Klik reactions yet, but we will eventually have it. So we can have, of course, an AI that is taught upon this knowledge. But I think that it's really important to understand that AI is not intelligent at all, because intelligence requires an underlying consciousness that we don't have yet in the computers. So AI is just a learning machine that has been trained, and then it has a number of layers of filters where the information that you add to the top of this filter stack ends up with a solution at the end, and then that is translated into a language that you can understand. So this is how AI works, and it works like that whether you are looking at molecular structures from crystal data or whether you are looking at ChatGPT and internet learning. So I think that we should look at AI just as we looked at the computer when that arrived to the scientific community. Another tool in the toolbox. AI is not going to take over from human beings, in my opinion. It can be competitive if we can manage to develop a consciousness or consciousness-like thing in the computer. We can get a self-evolving machine, which is evolving itself based on the interaction of the consciousness with all these tools that it has. Because AI is extremely good at programming. Programming is a quite exact discipline, and there's so much information out there for the AI to learn from. So in programming, AI is amazing, and it can actually program itself. So that means that you have the potential for evolution, and if you can just make something that's really like a consciousness in a human being, you can get a long way to a alternative species, you might say. But right now, AI is just a tool.
Yeah. Thank you. And so maybe if you are saying AI, I can give the right to ask.
Actually, my question is stolen. There was a kind of overlapping question. Maybe I can hand over the mic to my colleague here. He has another question. Kind of.
Professor, thank you very much for your amazing talk. And my question is about a little bit simple, but how do you structure your one day to stay productive and creative? I mean, how is your one day, a daily, from your daily life? Productive and for productiveness and creativeness.
How to be creative?
Yeah, how do you structure your day? I mean,
Okay. Well, that's almost automatic because my day is evolving around my students. When I come home from any travel like this one, I need to first use one day to repair all the instruments they have destroyed while I was gone. Because I have a policy of hands-on, so all the students, even the bachelor students doing projects, they can use the instruments. They need to learn how to do it, and then they can use it. And usually, this is very expensive. I just had a repair of a LCMS for 10,000 euros because one student did something wrong in the insertion of the probe of the samples. But I think this is very important for them to have this hands-on. It's much more productive in the lab when they do. So I have a lot of repair going on. How I plan my day depends on the students again, because in the night before I fall asleep, I'm concerned with all the projects that didn't work, which I told them what to do, and then it doesn't work. And they're probably right. I try, of course, as long as I can to tell that they are the ones that are wrong, but usually it's me. So I have to find alternatives, sit down with the students in the lab, talk through their projects, what they did. One very important requirement there is that they have actually investigated what they did. So if it goes wrong, what happens instead? That is an all-important question. So if they cannot answer that, I send them back in the lab to do the reaction again and investigate all the side products, everything that happens in the reaction.
Yeah, thank you.
Thank you very much.
And there's more question from the middle rows. Yeah, there is. Yeah, back.
I want to thank you for your sincere talk, firstly. And you said that we should teach chemistry to the young people and the children. As a young chemistry student, I want to ask that what do you know about the practice of science that you wish someone had told you at the very beginning?
What do I what?
What do you know about the practice of science, not the content, but the practice that you wish someone to tell you at the very beginning, so you could start the chemistry early or the science early?
Yeah, so you mean from when I was young myself?
Yeah.
The start of my interest for chemistry in the laboratory was in the lab with my master program, where I was doing some very nice reactions forming new kind of amino acids. And this was a great inspiration for me, but the real inspiration came with my supervisor for the PhD study, because he was the first one to introduce nuclear Overhauser enhancement in Denmark on superconducting magnets. So there you are taking your molecule and you can suddenly have an insight into the entire behavior of the molecule, what it's looking like, how is the electron density around each atom, how is the mobility of the molecule, how is the distances within the molecule in a particular conformation, and so on. What is the dynamics? All of that information you can learn from the NMR, and that was a real revelation for me, because suddenly NMR was not suddenly chemistry was not a foreign area. It was a real area. I could look right into it, see the interactions between molecules, and it became extremely exciting.
Thank you. And any more questions? There are ladies there. Yeah.
Hi, Professor Morten. First, I want to thank you for your inspiring speech. And you mentioned genetically modified Alzheimer's cells. And my question is, Do you think that this method could be applied to other diseases in different ways in the future, or is it feasible to mix biology and chemistry together?
Yeah, biology and chemistry and physics is all merging these years. So we are doing methodology development. Our laboratory is mainly working on developing chemical methodology. We develop instruments that can help us. We develop the chemistry reactions that can do things for us. And then we want to demonstrate that. And when we want to demonstrate it, we go to biology, where there's lots and lots of interesting things to use our methodologies for. So that is how we plan our research. I'm not planning Alzheimer research because I want to cure Alzheimer. I'm planning Alzheimer research because I want to show that I can make a recognition molecule that makes a difference. Right? So this is a completely different way of doing research than having a biological target that you're working on day and night. Probably you're more successful with your biological target if you're working on that directly. But I think that it's also important to have this more general development of technologies that can be utilized in a lot of different areas, and this is where we are quite strong. So I think that the genetic modifications, what we did was to use this amber stop codon technology, that's what you are referring to insert unnatural amino acids. This is actually done on many, many places, in particular in academia today, to investigate biological processes and to also develop alternatives to disease treatment. But it's also sort of kind of dangerous. So Peter Schultz, for example, has developed a bug that synthesized 5 new amino acids never seen before in nature. And he has the entire synthesis apparatus, the incorporation into the DNA and so on, in these organisms, and he has planted them in the back of his garden to see if they would be better survivors in our natural 20 amino acid organisms. I think that's quite dangerous, but probably we are lucky, and this is the optimal amount of amino acids, because we have ended up in evolution with 20 amino acids, right? Maybe 21 if you take selenocysteine as well. But 20 amino acids that produce everything, all functional molecules. And these 20 amino acids are selected to have the right functionality, and there's no more than 20 because of the energy balance. So in nature you have, just like a cash flow, you have an energy flow that has to be optimized with evolution. And that is the 20 amino acids, because it's very costly to introduce another amino acid in the system in terms of energy. So energy balance is causing, energy flow is causing evolution. The balance is resulting us with the 20 amino acids. So I don't think it's going to be a danger. Anyway.
Yeah. There is another question there.
Thank you, first of all, for coming down to Istanbul and for this amazing talk. It was great to listen from you directly, the Klik chemistry. I'm a member of Chemists Association, and I have an oligo house. I'm a custom oligonucleotide manufacturer at the same time. And who started me 17 years ago is your good friend, Carl Ross Peterson from Copenhagen.
I visited him just 2 weeks ago. I was in this.
I saw your picture on LinkedIn,
yes.
and I want a picture with you as well. But we use Klik chemistry as a post-synthesis conjugation. I'm wondering whether it will be at some point integrated to the synthesis process itself. But my real question is, apparently the biggest value of Klik chemistry is in vivo therapies, as we heard from you as well. What is your perspective about RNA vaccines and click chemistry in the future? It is obviously being played right now, and also I'm curious whether you have done something during the pandemics with coronavirus. Did you play with click chemistry and to do something?
We tried to make these recognition molecules for the spike protein, but we were not successful. We got very low affinities, so that was not a successful project. With respect to using the click chemistry in DNA, I think that is a very nice, already existing technology. But treating in vivo, you will need something which a click chemistry which is not toxic. And copper 1, of course, making all these oxygen reactor species is not a solution to that. So that's why Caroline Bertozzi actually developed the SPARC reaction, which is strain-promoted click reaction. And also, I think that the most efficient way today is a TCO reaction, where one of the two partners is used as a recognition molecule that goes to the cancer tissue, and the other one is a click partner that releases a cytotoxin when it clicks. So first you label all your cancer cells with this one click partner, then you have another one which is benign as it is because it's together with the other click partner, it's not toxic. Then it hits the cancer cells, and as it does, it will click and release the cytotoxin right at the site. I think this is a very promising methodology. I would love to actually precede the release of the cytotoxin with an import, and that's why we are working with cell penetration.
Yeah, thank you. For the also targeted therapies and also personalized approach, it could be really important to see the solutions. Yes, can we have questions? A few? Yes, there is a, yeah, there. Thank you.
So my question is about, as a successful scientist, successful chemist, what are your suggestions for young people?
Yeah, you mean when they fail, or what?
To be like you.
No, my suggestion is to do what you find is really important. Figure out what you're good at, figure out what is important for you, and do that. Don't do what other people think is important. Do what you think is important, because it's highly likely that other people will think it's important as well. And when you think it is important, you will work on it day and night, you will dream of it before you fall asleep, and so on. And this is your route to successful process. Also, I would like you all to be very concentrated on the moment in your research, not on the result, not on the final product, but on where you are actually right now. Find joy in doing a chromatographic column, getting a pure product out at the end, and going to the NMR machine and seeing spectra like I showed you here. This is such a wonderful feeling. And this is where you should put your efforts and your emotions, rather than the ambition to get a Nobel Prize, because if you have that ambition, you're probably not going to get it.
Yeah, live your passion. And with hardworking also, I think so. And so there is another question from there. Yeah, already there.
Professor Meldal, thank you very much for having us here. I'm a molecular oncologist with medical background. You already mentioned actually your technologies can be applied to cancer. I would like to know actually if we can reach you or a member of your team for future collaboration.
Yeah, so currently we have a collaboration with an American entrepreneur on this particular area. We are working with targeting specifically the cancer cells, entering the cancer cells and releasing in the cancer cells. So this is what we are working on, and of course, if that becomes a reality for us soon,
then we'll actually try to make a business out of it. But in the meantime, as academics, we are very open to collaborations.
Thank you very much. Because we are doing cellular therapies, and I can see a lot of your technologies can be applied to our research, I would like to reach you actually. Thank you very much.
There is another question from the youth, and so there will be, I think, the last two questions.
Hi. Firstly, I must say it's very exciting for me to talk to you.
For me too.
Thank you. And oh, I'm sorry. I forgot what I said. I'm sorry again.
Sorry.
What I'm about to say might shoot funny to all of you, but to be honest, it's not. It's my child dream, and it's my very, very big child dream. You know, you are very respectful person for us, and you have won Nobel Prize, and it's chemistry Nobel Prize. It's very big thing to me and us, I want to say. And it might be ridiculous, but can I touch your Nobel Prize with only my one finger?
Hey!
Thank you so much. Don't be worried, my hands are clean all the time.
I'll take a selfie with you where you hold the Nobel Prize.
Thank you so much. It's very my big dream. Oh, I can explain myself because thank you.
Yeah, I will.
I can't explain how I thank you.
You will hold this. You will hold it like that, and then you will have a picture.
Thank you. Thank you so much. You are my big one. I can't speak. You are one my biggest idols.
You are a very courageous person. You will get far in life.
Thank you.
Thank you.
Thank you so much. Aslında böyle kişisel bir soruyla programın profesyonelliğini bölmek istemiyordum, fakat bir daha şansım olur mu hiç bilemedim. Çok teşekkür ederim. Sizden de özür dilerim.
I think that
Very emotional.
it's the mission completed, I think. And so, yes, hocam.
Yeah, exciting. Yeah, after such a nice thing, and I think you are a superhero of many people, and when I heard that you like also science fiction, so my question is about, chemistry. If you would have a super, power, what would you like to have? Because I have one, I will tell after yours maybe. I would like to have at least, for example, NMR, like the eye.
My superpower is a really simple thing. When I was a kid, I always had this nightmare of falling. You know, when you are sleeping and suddenly you fall and you wake up like that, And then I learned to control that. So in my dream, I would start falling and then I would turn around, I would start flying. So this is my superpower that I would really like to be able to fly across the world.
So you were Superman. But if we consider chemistry, and we use several spectroscopic methods, and there are lots of powerful methods, of course. And if your eyes could be one of them, what would you like to pick?
So I would really like to learn exactly why we cannot understand chemical recognition. So we are very close with our computer programs to be able to compute molecular recognition.
Thank you.
But we are also very far away because sometimes you have an antibody and an antigen. They come together and we can calculate an affinity.
And this affinity can be a million wrong from what we measure. And this is unprecedented in science, that we are so wrong in our calculations. And I think that there's something hidden that we have not taken into account yet, which is actually inherently understood by AlphaFold, the AI that understands chemical structure. So we don't know what it is that controls the outcome of AlphaFold. When we ask a particular sequence, it folds it up and gives us a structure. There's a lot of molecular recognition involved in that. It's not directly calculated molecular recognition, but it's all the statistics from the database that is included there. And these statistics inherently contain also the information we don't have. I think there's something about antibonding orbitals and bonding orbitals interacting in the molecular interface. And this becomes, of course, more and more complex as you go to larger interactions like an antibody with a protein surface or something like that. And we cannot calculate it accurately, and this is a real problem for me. So I would love to have the superpower to understand that.
Great, thank you.
There will be last two questions from the Just Youth audience I would like to have. There is one there with microphone, yes. And then
Hi.
I think you can ask your questions also, but yes.
Hi, Mr. Morten Meldal. First, I want to introduce myself to you. It's a very pleasure to meet you. My name is Meldal. I'm a high school student at Kim Vocational Technical
Can you just
and
take up your hand, because I don't know where are you sitting?
Yeah, it's on the corner.
All the way up there, okay.
Sorry. I'm a high school student at Kim Vocational Technical and Anatolian High School. I have two questions. Can I ask both?
Yes.
Question? My first question is, if you were a high school student currently studying chemistry, would you take any initiatives? What kinds of activities would you participate in? What would you do in this age of technology? What would your move be?
Yeah, so I would, of course, I'm very interested, as I told you, as a young person, in nature, but we didn't have the internet, we didn't have all the tools that you have today. I would use those tools to all the time find new information about the things I'm curious about. I would also maybe take some more popular, easy to understand books about chemical reactions. Chemical reactions are really, really important in chemistry, understanding the way that electrons interact and how, and the earlier you get this kind of understanding or get this, you also get inspired by understanding it. So the understanding in itself is an inspiration for chemistry. I can feel that also when I have my course, the Meldal course. The students really love this, oh yes, that's how it works, feeling. You know, this is really, really important feeling that you can understand something as great as how the molecules interact and how they react with each other.
Thanks for answering. My other question is, click chemistry is a brilliant way to synthesize complex molecules. Do you think similar click mechanisms could occur naturally in the interstellar medium or nebulae? How can your methodology help us understand the chemical origins of life in space?
Yeah, so I think life is not originating in space. I think life is originating on Earth. It's originating by having the right conditions where you get exactly the kind of kinetics that you have had in evolution for molecules to interact and react with each other on the right time scale and so on. So life is not coming to Earth from outside. It's inherent, living inside the atoms. So it's created by the conditions that we have here on Earth. And if we look out in the universe, in the interstellar region, there is 2 trillion galaxies in the visible universe. Each of these galaxies on average contain, and this is a huge number, 200 to 300 billion stars on average. So if we calculate how many stars are out there, it's almost like Avogadro's number, and that's 10 to the, yeah, I cannot really calculate it. It's what, 3 times 10 to the 14th or something like that? I don't know. It's a very, very large number, and each of these stars has a potential to have conditions on the planets around it, like here. And when the conditions are there, it will happen.
Yeah. Thank you for answering. Pleasure to meet you, and pleasure to you for answering my questions.
Yeah,
thank you very much for asking them.
Last question. And so, as you can see, there are many questions.
Yes.
Maybe we will have time.
Yeah, I have all the time of the day,
yeah.
so...
yeah, today we... Yeah, thank you. So, during afternoon, there will be programs, and so Professor kindly is going to be here all day. And so for this reason, to proceed the program, unfortunately, I couldn't give you a voice. But there will be opportunities, and so we will have photos and some other things. And so it's going to be the last question from the youth part.
Hello?
Yes.
Hey. First of all, thank you for the lecture. And my question is about peptides and their potential medicinal applications. So, as you may know, peptides are also used for muscle recovery and for muscle injury treatment. So, do you think that click chemistry could be used for producing peptide-based treatments for muscle recovery in the future?
I'm not sure about muscle recovery, but I'm sure that it will be used in drugs. It looks like it's completely benign in the immune system. So the triazole click reaction seems to be not affecting the immune system, the immune response. And I think that you will see a lot of peptides that contain this, but not only peptides, also combination of peptides with other kind of entities. So you have, the Wegovy is a good example where you have this long fatty diacid sitting on a peptide. It connects to albumin in the blood, and that is transported around and released slowly, and then you have this wonderful magic bullet effect of the GLP-1 of that peptide. So I think a lot of peptides like that will be produced in the future using the same kind of system. And there, click chemistry, I know Novo Nordisk have a huge department of click chemistry, actually headed by my former PhD student, Christian Tornø, who was also a big contribution to this Nobel Prize. And he is working on making click chemistry part of their peptide drug program.
Thank you for answering.
But I cannot answer you on the muscle regeneration or bone regeneration. I know that there are people working on stem cell research for bone regeneration. That's not the same, but they are using a peptide matrix, immobilizing stem cells in that, and then inserting that into completely fractured bones to regenerate bone structure.
Thank you for answering.
Before the closing ceremony, as you can see, many questions, but it will be maybe answered at the stage part, so we will have time, don't worry about that. And so, last question is going to be from my side. How do you find us? And
Turkey, I love Turkey. It's been a wonderful 2 days. I mean, in Ankara yesterday, me too, and now today here. But I would need tomorrow as well, because tomorrow we are going around Istanbul, seeing all the historical sites together with Erhan and his wife, and this is going to be
amazing.
Amazing.
And
we are going to the hammam.
Yeah. You have another house here, I think, as you can guess. So anytime you want to see you here, and there are really lovely students, and so you have to. We hope to again see you under this building. It's a great pleasure for us, and also I would like to also express my sincere, thanks to all audience, because your attendance and also contribution is valuable.
That's
makes.
for me to thank you. Thank you very much for coming.
And now we are having the last part of this program to kind of to collect memories together. And for this purpose, I would like to first invite my dearest rector, Professor Hacı Ali Mantar, to the stage. And so there will be a kind of the small gift and photo ceremony. For all audience, please stay for a while because we will have a photo together. And thank you. Yes. May I?
Thank you very much.
Thank you. Stage is yours.
Thank you. Thank you for having me.
Microphone.
Wow. Thank you.
I think you all know, thought that the computer system didn't work. Actually not. The computer system was working, but the presentation was invaluable presentation. The computer system cannot understand. It cannot do it. Later, our vice rector tried to pass the slides, but since she didn't have a deep knowledge about the chemistry, it didn't work again. After that, the expert comes and it works. That's why. Thank you.
This is one of the art pieces made by the Ebru. Is it a kind of the Turkish art? It's
Yes.
called as a chini. And so this is our logo. It's the butterfly as a technical university.
It's very beautiful. Yeah?
And so there is a very nice.
a lot of
And because of the biodiversity, it's really protected under this campus.
Yes,
And
thank
so
you.
I also would like to, And also I would like to invite Professor.
He's very popular. He's very popular.
Yes, are the dean of the science faculty. To present a commemorative gift to Dr. Erhan Atıcı. He's running.
Thank you for the invitation.
This flowers to your lovely wife.
Oh, yes. My wife is sick, so she is not here today. She's usually joining me on all of these events, and we have a very good life together. So this is for her. She'll be so happy.
Now may I have invite the members of the organizing committee in Fokutan, Burcu Dedeoğlu, Tuğçe Gevrek, Kivan, and all young researchers who work hard for this organization. Immediately, may I have all? Yeah, thank you. Thank you. For the photo. And the researchers, and deputy vice rector, vice dean, center, And now I would like to invite all chemistry department and vice rectors, deans, and the students, Sarkan hocam, Oğul hocam, Yusuf hocam, Orhan hocam.
coming back. I'm coming back. Coming back.
Take the students here also on this place. Odanı çektim ben biraz. Ya Sadi hocam. You can stand here also, please. Use this area. Yes. Please stand here and all faculty members.
Now
Please use the spaces. Be careful. Tamam. And at the end of the event, please, we will go to outside, and on the stairs there will be another memory photography together. Bu fotoğrafı çektirdikten sonra dışarıya çıkacağız. Dışarıdaki açılış kapısındaki merdivenlerde fotoğraf çekiyor olacağız. Fotoğrafçılara bir de izin verebilirseniz ön tarafta oturanlar için rica edeceğiz. Teşekkürler.
Dur, bekle.
Hocam sizi de çekiyoruz, öyle düşün. Lalecim bizi böyle de çekin.
Dur.
Okay.
Sorry. Okay.
Now we are ready. Kapının önündeki merdivenlere doğru gidiyoruz. Orada da fotoğraf çekilecek.
Hey, birisi çekin. Yanına durun, birisi çekin.
Can you take the medal out?
The medal.
Çocuklar, bizden bekliyoruz bak bunu. Süper çek. Hocam